Episode 5 I started the DNA reset. Episode 5 The role of epigenetics in regulating gene expression. Welcome to the 5th episode of our series on DNA and its repair and regulation mechanisms. Having discussed the various mechanisms of DNA repair, we now focus on a fascinating and highly influential field in modern biology, epigenetics. In this episode, we'll explore how epigenetic changes affect the activation of genetic traits without changing the DNA sequence. This process has profound implications for organismal development, environmental adaptation, and disease pathogenesis. What is epigenetics? Epigenetics studies heritable changes in gene function that do not result from alterations in the DNA sequence. These changes can affect how active a gene is without changing the actual genetic sequence. The primary epigenetic mechanisms are DNA methylation and histone modification and interfering RNA. Main epigenetic mechanisms: DNA methylation. Adding a methyl group to cytosine in DNA can suppress gene activity. This modification is essential for embryonic development, X chromosome inactivation and genomic imprinting. Modifying histones. Histones, the proteins around which DNA is wrapped, can be modified by adding or removing chemical groups such as methyl or acetyl. These changes influence chromatin structure and regulate the access of transcription enzymes to DNA, activating or repressing gene expression. Interfering RNA. Small RNA, such as small interfering RNA, mRNA, and small interfering RNA, siRNA, can control the expression of genes by inhibiting the process of translating them into proteins' messenger RNA messages or degrading them. This mechanism is crucial for regulating gene expression and cellular stress responses. The influence of epigenetics on the well-being and illness. Epigenetic changes can profoundly affect organisms' health and development. For example, changes in DNA methylation are implicated in cancer development because they can activate oncogenes or inactivate tumor suppressor genes. Also, certain hereditary diseases, such as Prader-Willi and Angelman syndrome, result from dysfunctions in genomic imprinting. Epigenetics and environment. One of the most exciting aspects of epigenetics is the influence of the environment on epigenetic changes. Environmental elements like nutrition, tension, and chemical exposure can alter an individual's epigenetic profile. These changes can affect the individual concerned and be passed on to subsequent generations. Conclusion: epigenetics opens a new horizon in understanding how genes are regulated and expressed. This not only adds a new layer of complexity to what we know about the genome but also offers new perspectives for developing personalized therapies and treating complex diseases. Thanks for watching this episode. We hope to see you again in the rest of the series, where we explore other fascinating aspects of molecular biology. See you in the next episode.